welcome you all to the 11th session of Cerrahpasa Neuroscience Talks. Today, we have the great privilege of hosting an exceptional scholar, an influential expert in the field of neurophysiology, Professor Giacomo Rizzolatti. Our focus today will be on the intriguing topic of the mirror mechanism. As we eagerly await the talk by Professor Rizzolatti, let us first acquaint ourselves with his background and his accomplishments in the field. His alma mater is the University of Padua, which he graduated from in 1961. He also obtained his specialization in neurology in 1964 at the same university. He is an elected member of the Academia Europaea, National Academy of Sciences, and Royal Society. He is also a past president of the European Brain and Behaviour Society. He was the 2007 co-recipient with Leonardo Fogassi and Vittorio Gallese for the University of Louisville Grawemeyer Award for Psychology. Since 1975, he has been a professor of human physiology at the University of Parma, with a short break of a visiting professor position at the University of Pennsylvania. Professor Rizzolatti is internationally renowned for his pioneering research on the mirror mechanism, a basic neural process through which the brain transforms the sensory observation of others' actions into corresponding motor representations in the observer. This line of research has had a profound impact on neuroscience, psychology, and cognitive science, offering new insight into how we understand others, not through abstract reasoning alone, but through direct and embodied neural mechanisms, which we are delighted to hear about from him today. Professor, welcome again. The floor is all yours.
Well, unfortunately, the professor has dropped.
Dropped again?
Yeah.
I wonder when.
I believe he will come back soon. So we're just going to wait just a couple of seconds more. Yes, here it comes again.
I'm glad our participants are acquainted with him now.
Okay, we are here.
Now we are connected again.
We're glad. So, as we all know, Professor Rizzolatti is internationally renowned for his pioneering research on the mirror mechanism, which we are so excited to hear about that from him today. So, Professor, welcome again. The floor is all yours now.
Thank you for your words and good morning, dear colleagues and dear students. My talk will be about the mirror mechanism. Here is the definition of what is the mirror mechanism. What I want to do is to speak about the mirror mechanism and also on how we discovered it. So the mirror mechanism, as written on the slide, is a basic neural mechanism that transforms sensory representation of others' actions into motor representation of the same action in the brain of the observer. We discovered it several years ago, and our discovery started with anatomy. So at the end of last century, there was a lot of discussion about area 6. That's what you see here. It's the map of Brodmann that everybody of you knows. And area 4, it's unitary. But area 6, there were discussion if there are several subfield or there is a unitary aggregation of. So we did an experiment on monkey using different techniques, and the result showed that indeed. Sorry? You cannot hear me?
No, everything's fine, Professor. There was a mishap with a participant. You can continue.
So if there is any problem, tell me. I will try to. It's okay.
No, it's not a problem with you. It was with Zoom, but it's solved now. You can continue. Sorry.
So, as I said before, we were interested in learn if area 6 is unified or it's made by different fields. And the results are shown here. Here is the brain of the monkey. The motor cortex is area from F1 to F5. And what we found is that indeed area 6 is very heterogeneous. Today, I am interested in one area, area F5. So anatomy first, then physiology. Once we discovered that area F5 is different from the other areas, we started to record from it. We used the records of action potential. It's still probably the most powerful way to understand what a certain neuron does. So you probably know that action potential are the language of the brain. Here it's action potential recorded from a nerve, but the same is true if you record from visual or motor cortex. Well, what was our original idea, which I think was a winning approach, was to use an ethological approach. Instead of looking for force, kinematics, and so on, we play it with the monkey. We give the monkey some food, we throw it away, we look at how she grasps it with two fingers or the hand, and so on. And the results were very interesting. First of all, we discovered that area F5 has visual properties. The first thing we discovered was look at this figure and essentially at the last one panel, object fixation. With our surprise, we found that a premotor neuron was discharging when the monkey fixated this object, this specific object. And of course, when the monkey was grasping the object, was the same. Here is the visual response, and here is the motor response. So this type of neurons, similar neurons, were recorded in parietal lobe by Sakata and his co-worker. So we call it this neuron canonical neurons, neurons which fire in response to particular object. And Murata, who was a co-worker of Sakata, worked in Parma for a couple of years. But the great surprise was the following. At a certain point, we found neurons. We found when the monkey grasped the object, B, here there is nothing surprising. But also when the monkey observed the experimenter grasping an object. It was really something amazing. How it's possible that a motor neuron fire when the monkey observed a specific type of movement. Here is one of our protocols. What you will see here, you will hear tack, 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 tack. That's the sound of action potential. And you have to correlate this tack, 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 tack with the action you will see. Grasping neurons.
Fanno un altro così?
Up to now, no surprise.
Feeling.
Grasping. Same neuron. That's mirror effect.
Ancora?
The same neuron you saw before firing during grasping now fires when my coworker grasps the object. And here it's interesting because it's grasping. Even grasping with the mouth is effective.
Gli stimoli che riconosce.
You see, I repeated several times in order to show you that it's a very strong effect. It's not related to statistical analysis. It's not something that one can find because make some complex manipulation. It's just a neuron there. I think even a child will say, yes, that's neuron fire both during action and during action observation. However, while my co-worker were very happy and say, let's send immediately to science. It's a great discovery. I was a bit scared because to see something of this kind, it could be some kind of artifact. Maybe the monkey is moving something and we don't see it. However, there were some neurons which convinced me that it's so. After 4 years of control, that indicates how I was scared to do something wrong, we sent it to brain and it was published in 1996 and was called mirror neurons. From that time, we use the English name because we have many foreigner in our lab, and so the English was the official language. So we call it mirror neurons rather than neuroni specchio, that will be the Italian word. And the community of neurologists and physiologists accepted it with pleasure. Ramachandran is a famous neurologist of Indian origin who graduated in Cambridge and then worked as professor in California. He wrote, at the beginning of this century, there was a survey. They asked many famous neurologists and physiologists, what is the most important discovery that will influence the next years? And Ramachandra wrote, mirror neurons will do for psychology what DNA did for biology. They will provide a unifying framework and help explain a host of mental abilities. Okay, now great success among neurologists and physiologists. Psychologists were a bit skeptical. They say, well, but that's monkey. Are you sure that also humans have this system? And of course, it was interesting to find. We were lucky that in a university close to Parma in Milano, they opened in those days a PET lab, positron emission tomography. You know what is positron emission tomography? It's a procedure that measures metabolic activity of the cell. So if a cell is very active, there is more activity. And if you inject radioactive glucose or desoxyglucose, you can see, thanks to some technical procedure, the area which is active. Using PET, so we went to Milano and we injected in the subject desoxyglucose, radioactive glucose, and the results were here. The yellow areas correspond to area 5, it's ventral part of area 6, and partially also area 44, that's interesting for language. And also was inferior parietal lobe. In the monkey, we didn't know yet that, but subsequently with Fogassi, one of my co-workers, we found that the same is true also in the case of monkeys, not only humans. Here was a crisis. In Italy, there was a kind of phobia of radioactive material. After a while, many students said, no, I don't want to be injected with radioactive material. It's dangerous. I may have a cancer. You say, no, listen, it's a very small amount. Anyway, it was difficult. Fortunately, in Germany, we have collaboration with Düsseldorf and Jülich, and we use it, German student. And you can see the results were very interesting because here you can see that there is a clear somatotopic organization. When you look to movement made by feet, you have activation here, but for the mouth, you have the ventral. So you probably know this map, it's classical map, somatotopic map by Woolsey and Penfield, and mirror neurons show exactly the same. Mouth is here and the feet are located dorsally. Okay, at this point, we have a big fortune. The big fortune was fMRI. Functional MRI is a technique, you probably know, that determines the brain areas active during the execution of tasks. And it's based not on radioactivity, but on physical properties of magnetic field. fMRI does not scare anybody. So in some years, there were many experiments on mirror neurons using fMRI in humans. And here it's a review article. These are data from a review article made by Carl Zillers, a famous German anatomist, based on 90 experiments with mirror neurons. And the most important result you can see here is the presence of an activation of the circuit which go from the parietal lobe to premotor cortex. That's the well, anyway, you saw it. I don't understand why. Now I go back. Now it works again. So I finish it with this German experiment or German survey, which indicate which area are active in humans in the mirror system. And here is, sorry for that. Here we are. Now the problem is, what is the function of mirror neurons? At the beginning, we thought, maybe in a very naive way, that the fundamental aim is imitation. And it's true. In humans, imitation is based on mirror mechanisms. The experiment I carried out in America with Marco Iacoboni showed that that's true. But ethologists, in the meantime, discovered mirror mechanism also in birds, in monkeys, in rats, in mice, in bats. So it's something very diffuse. Furthermore, ethologists told me, look, but mice or bats are unable to imitate. So your proposal is valid for humans, but it's not valid in general. Mirror mechanism should be a mechanism which is older than the capacity to imitate in the proper sense. At this point, what could be the other hypothesis? The other hypothesis was that it's important for understanding the action of others. So when I go in a bar and I see somebody taking a cup of coffee, in my brain there is a similar mechanism and I understand directly that the guy is grasping coffee. So that's the basic mechanism which is present in all the species that you see here, birds, monkeys, rats, and so on, because it allows you to understand immediately what the other person is doing, not using complex cognitive processes. So we did an experiment to see which are the actions we understand using the mirror mechanism. So we did an experiment, fMRI experiment, in which in one series of films, the subject lying in the scanner saw grasping with the mouth made by a boy, made by the monkey, and made by the dog. So everybody was doing the same action. And the results are here. If you look at this red spot, they are exactly in the same way. So I am understanding the dog biting or the monkey biting exactly using my motor program. What I said about the bar and the coffee is true also when I see other animals, not only humans. In the second part of the same experiment, we presented action which is typical for a given species. Thus, for example, the man was looking at a person which was saying something by lip reading without sound. The monkey was making lip smacking. conciliative gesture, which say, we are friends, don't do the war. And finally, the dog was barking. If you look now at the area which are active, they are completely different. For example, barking of the dog is not understood by some motor program. We don't have a motor program for barking. I don't know. I can say bow bow, but it's not what the dog feel when he's barking. Instead, we recognize it because there is a visual activity, and of course, we have subsequently some logical inference about that. We call that understanding others from the inside. So we have a basic mechanism, which is the mirror mechanism, which allows us to understand others from the inside, copy their action on ourselves, like the guy in the bar, but we have also the possibility to understand complex stuff by reasoning and so on. Now, I gave a talk like that, and the gentleman came close to me and said, listen, beautiful data, but I know there is another aspect in action that you never mentioned. This is how the action is made. This guy was Daniel Stern, a famous psychiatrist and psychologist. He wrote a very interesting book about the development of action in children. And he said, why we don't do an experiment together? But before, I want to show what he means by form of an action. So an action has a content, grasping, and how it's done. You will see here, he called it, Daniel Stern, this aspect vitality forms. Now look here. The boy said, be quiet, very gently. The same action, be quiet, but the way in which it's done is completely different. He's rather rude. The first boy was gentle. Here is a transitive action. The girl is giving the crackers to the boy very gently. Certainly, it's different, the relation between the two. I don't think this couple will last for long if they are so rude one with the other. Okay, now I think you understand what is vitality force. Vitality force is how an action is done. The action is the same, can be done in different way. We did the first experiment with Daniel Stern in Parma, and it was a very simple experiment. We showed two action, and the two action could be similar in terms of content or in form. One, they were similar in terms of form, regardless of content. There was an activation of this yellow spot here, which is the insula. You know, the insula is the structure in the depth of the brain that you can see if you open the two sides of the Sylvan fissure. Well, subsequently with Di Cesare, one of my students, we did a more complex experiment. So we presented action which were gentle, rude, there was a control, and we have three conditions. One condition was observation, one was imagination, and the third was execution. And the results showed that in all these three conditions, the focus of the vitality form was in the insula. This yellow spot is in the insula, and it indicates also that also for vitality form, there is a mirror mechanism. The same area which is active when I imagine or observe the vitality forms, it's active also when I execute vitality form. And here is the insula. I suppose you know already, you already learned it in anatomy, that's the insula. The part which is active in vitality form is the rostral part in its dorsal sector. That's something very recent. Together with an English famous scientist, Carl Frischt, we used this dynamic causal modeling to see what happens with vitality forms. DCM is a method used to estimate the coupling among brain regions and the changes in coupling due to experiment procedure. In other words, we give a stimulus and certainly in a certain area modify. Then using Bayesian statistic, we see which part of the brain is also modified. So we can see in this way the coupling between different brain regions. Here is a recent last year published experiment with Freestone. And now I will show you the results. Look here. Here is action observation network. If there is action without vitality form, just I move the hand like that, the activation starts in STS, which is a higher order visual area. It goes to the parietal lobe and to the premotor cortex. That's the simple action observation network. However, if I do one of the action I showed you before, it starts again from STS, but then it moves not only to parietal lobe, but also to insula, and there are two circuits which converge to premotor cortex. So when we do an action with vitality form, we have the insula which became active. That's in case of observation, but the same is true also in the case of action execution. Now, emotions. Emotion is an interesting chapter which needs some definition. I use the definition which classical goes from Darwin. It's not emotion like people use, today it's very emotional or something. That emotional, it's very well defined by Darwin and later on by others. Emotion are complex reactions that people experience in response to specific events of situation. It's a very short-lasting effect. And the basic emotion, as studied by Paul Ekman, are disgust, fear, pain, anger, joy, and surprise. So it's not something that, I am thinking that this guy is bad and therefore I am in good mood, in bad mood. No, they are very well defined, are typical behavior of human and animals, which are written here, disgust, fear. Disgust is extremely important because it allows you to save not only yourself, but also the other people. Let's imagine that I go in a place, I eat something, and then I vomit. The tribe or the other people will never go there to eat because it provokes war. So in this way, with showing disgust, I am able to save not only myself, but also the other fellows of my tribe, of my group. So we did the experiment using odorants. Odorants are extremely strong stimuli in eliciting emotion, especially disgust. So we have two possibilities. Use natural stimuli, and the best odorant which provoke disgust is rotten eggs. Rotten eggs is excellent because they determine disgust in human, in men, in women, in boys, in everybody. And then we use social stimuli. Social stimuli means that you express disgust, but you are not disgusted at all. You just show that you don't like something. Here are the results. The first result was just normal disgust due to rotten eggs. It's this rostral part of the insula which is activated. That's not our original finding because before us, a group at University College have already described this disgust activates this part of insula. The original part was the following. Here we showed to student movie of people showing different emotions. The first one showed disgust. These are actors of Marseille theater, as a matter of fact, so they are expert in doing that. This guy is happy, and the nice girl is just doing nothing. It's neutral. No. So what are the results? If you see here, the most interesting result is the following. You see these white spots? These are white voxel which has activated both when you feel disgust because you have something disgust in your nose, and when you observe this guy. So in other words, it's a mirror mechanism. I map his disgust, his expression of disgust on my basic structure for disgust. Well, up to now I have spoken a lot of brain imaging, but there are some limits of brain imaging. One limit, which is really very dangerous, it's called correlation errors. You do a fMRI experiment and you see that area A is active. Well, you say that's the area for pain, but it's true. Or there are many areas for pains, or pains is something more complicated. Just to give you an example, how we can overcome correlation errors. By using another technique, not fMRI, but just by recording from the brain of people. It's called stereo EEG, and it's used in order to localize epileptic focus in epileptic patients. It's a rather complex procedure, and we were very lucky because in Milano there is one of the best centers in the world. Another very good is in Paris. How we can know where is the epileptic focus? Well, first of all, we do a resonance. That's a structural resonance. Then we use specific electrodes which allow us to record from several points. We do angiography. These in blue are the vessel. And then we insert, the surgeon insert in the brain many electrodes. These red points are where in the brain of a patient has been inserted the electrode. And the electrode, as I said before, was electrodes with several recording points. So if we have 16 electrodes, if each one have 10 points, we have 160 points of the brain that we can explore. Here you show why correlation errors are very important. This is a review by Prosic reported a large number of function of cingulate, MCC, its middle cingulate, ranging from feedback to pain, salience, action reward. Now, can you believe that this small region here, the blue one, which is the middle cingulate, can support all this action? Of course, there were some doubt. Now you will see what happens when we use stereo G. Now I will stimulate electrically exactly that port. You will see the behavior of the subject.
Uno, due, tre, quattro, cinque.
Che c'è? Che c'è? Cosa è successo?
Tu vuoi andare via?
Cioè?
Mi hai sentito che vuoi andare via così?
So after stimulation of this area, the boy said, I want to go away. He had to go away. The doctor said, why? I must go, I must be going, I must be going. Why? Because it's an area which produces anxiety. When you stimulate this area, he became very anxious. And that's why all these symptoms that Prozik described are true, but are not related to a center for pain, but is related to a center for anxiety. Now I will show another experiment. This time we will stimulate the anterior part of the cingulate, this red area, PACC. Here is a girl, epileptic, that will be operated in few days, and the surgeon stimulate this red area I show you.
Lavoro coltello, maestra, ferro, gatto, arancio, pulsante alto. No, è tutto un attimo di...
Da ridere?
Sì.
The doctor said, you are laughing. Yes, she said. And then the doctor will ask you, but why? I don't know. It's something inside me. This
Lavoro coltello.
guy here, it's more introspective.
Zanzara, fuori, pane, schermo, borsa, albero, nuvola, cancello, laccio, acqua. Zanzara, foglio, pane, schermo, borsa, albero, nuvola, cancello, laccio, acqua. Mi viene fuori, così non sono io.
You see, he is very introspectively clever. He said, it is not me. It's something inside myself which compels me laughing. And that's true, because there is no reason to laugh. They are all patients in the hospital. You don't laugh. You are sad usually. You expect operation. And they are compelled to laugh because we stimulate the laugh center. And in the other patient, the anxiety center. Now, with Fausto Caruana, we decided to see, but for laughing, there is a mirror system. This guy said, I am laughing without reason. But if we show him a person who is laughing or a movie which is funny, what happens in his brain? And the result was very clear. This red area indicate the activation in that precise point where stimulation determines a laughing when he sees somebody laughing. When he sees somebody which is neutral or it's sad, there is no activation. So also for laughing, other people demonstrate that for fear there is activation of the amygdala. But anyway, these data clearly show that for emotion you have the mirror system, and the mirror system exactly allow you to understand the emotion of others. Now I am almost finished. At the beginning even of our research, we said, but what we can do with this discovery, there is a practical application, and we call it action observation therapy. What means it? Imagine a person who has blocked the arm in this position because there is a broken arm. For 20 days, he is in this position. When the cast is taken away, he can move, but he moves very poorly. Why? The old orthopedics say, well, because the muscle became weak. It's not only that. It's because the motor program of moving, it's sleepy, it's weak. It's not good. But if we present to this patient movement well done by himself or by another person, this decrease of motor program disappears. And when you put away the cast, the movement became normal. I don't know if you saw patients which have hip problem. And the people with hip problem walk in a very strange way. So they have this parasitical motor program. When you put away when you substitute the bad hip with a good one, he walks very badly because the parasitical, the wrong motor program is there and compel him to work in a bad way. But if you show him how to work well before surgery, when you operate, he recovered immediately very well. So this technique is called now action observation treatment or action observation therapy. It's used here. It's also a very good paper many years ago made. It's the first one on deficit after stroke. It's the same thing. When you have a stroke, you have a period in which you are completely paralyzed. Then there is recovery, and if you show the real movement, then the things are going very, very well. Now, motor rehabilitation is used in many diseases, in paralysis, in the Parkinson disease, in the multiple sclerosis, in hip prosthesis. Now about hip prosthesis, I already mentioned, but in Milano, for example, they use to show to the people who will be operated by hip prosthesis the normal walking before surgery. So after the surgery, they recover in a few days. Orthopedic disease is obvious. Well, thank you. I am finished.
Thank you, sir, for this delighting presentation. So, if you'd be okay with it, we can proceed with the question and answer part.
Okay.
All right,
A
then.
few questions, okay.
All
Not
right, a
many.
few questions. So, we'd like to start with a question that was directed at us to you prior to this. And it's an interesting one, so I'd like to ask that to you. One of our participants asked, although our main topic is how we understand each other via mirror effect, I would like to ask if there is any situation where this phenomenon causes misunderstandings in humans, such as yawning, where the other person may mimic it despite not being tired. So the other person may think we're bothered, bored, tired, when it's only true for one party in a discussion. As someone with Tourette's syndrome, I have come to notice other people with tics, or sometimes the other people repeating my tics as a joke can make my tics even worse. Is this also linked to mirroring? One participant asked.
Well, if you observe somebody doing tics, you have a strong tendency to imitate him. So I think it's a very bad joke to to show tics to somebody who has already the tic disease. I would recommend not to do that,
Yes.
that kind of joke with him.
All right, now the professor has accepted like one or two more questions, I think. So you can either write your questions in the chat or raise your hand, which would be even better. Yes, please.
Yes, please.
Fatma Zehra.
Hello.
Yes, I can hear.
First of all, thank you for coming and sharing all this precious, valuable information with us. And then also thank you to the committee. It was really exciting to listen to your presentation. So while I was listening your presentation, I had this understanding that comes from Panskip, who is the father of social and affective neuroscience, which I'm interested as well. I know it is different area of expertise, but I wanted to ask, and I was thinking while I was listening, since play, which is the actually the social frame of our actions with others. So if we frame the action or social interactions as a play, which was actually emerged in early mammals, roughly like 200 million years ago, evolutionary. And many species, especially mammals, we show these forms of action mirroring or timing-based social play. Do you think that the mirror mechanism originally evolved to read the pattern and vitality of play actions rather than action themselves? So what does this imply about the evolutionary roots of human social understanding as play? One question is that.
Not completely. I think the mechanism is, we don't know how it turned on, but certainly it's a mechanism very useful because you don't imply the frontal lobe, reasoning and so on. And it can be possible also in animal very simple, like rat and mice. There is a recent very beautiful paper made in Stanford about anger. When a rat is angry, it's activation of the hypothalamus, and when the animal sees two fighting, is the activation of the same area, which indicates it's very ancient, but it's also very simple in a sense, because you don't need reasoning. We understand many things by reasoning, we humans. But animals can understand many things just by observation. And that's the big advantage of mirror mechanism.
Just my last question. Then how we can relate the activation in insula, which is in the recent publications, it's all about as well, partially about the self. When we deeply...
About what, sorry?
Self. When we deeply internalize self, when we start thinking
Yes.
about our own actions, thoughts, mostly it is seen as well in default mode network activity. So then how you can relate this interesting pattern, which frontal areas are not excluded.
No, I think you have been very careful because insula is sometimes considered as the insula, but the insula it's a lobe and it's completely different. The rostral part, there is very good papers indicating which part. First of all, there is the anterior insula and the posterior insula with the sulcus in between. And all data we have is on anterior insula. And if you go very, very front, in the frontal part, this insula became very cognitive and related with prefrontal lobe. So I think that to explain some finding of the insula, one must know which part is it and which is the connection of that part with the other part of the brain. I'm sorry if I have not answered completely.
No, you just...
Insula somehow worries me because people speak about insula or insula epileptic seizure, but it's a lobe. It's not just a small area.
You are just sending me to different direction to find it away. Thank you.
All right. I see some questions in the chat.
If
Professor,
there is a
one of
last
our...
question, I will be happy to answer, but the last one.
All right, then. Let me choose the question. This is an interesting one. One of our participants asked, would mirror neurons be activated in an individual who was born without a hand when they observe another person grasping an object with a hand? Is there a possibility?
I think it will recognize it because, as I show you, even grasping with the mouth is active. So we have multiple neurons which are activated when you observe grasping with the hand, with the mouth, but also with the feet. So it is a generalization. The question was very short. I can do another one if you want.
All right, then. It will be our pleasure. And here's a question. They said that this participant has a student with cerebral palsy, and she says that his mobility is getting worse, and she would like to know if AOT would maybe help her patient, or in what ways.
I have not understood. What is the question exactly? If who improve?
So the participant asking the question has a patient with cerebral palsy, and
Yeah.
she is wondering if his moving mobility could be helped with AOT.
Absolutely. AOT can help it very much because it enter inside the brain, which is in a sense sleepy. Of course, it's destroyed, it cannot be. But if you think about the pathology, there is a part of the brain which is destroyed, but around there is edema, and the part of this brain is not working well. If you don't do anything, this part disappear. But if you do action observation therapy, this part wake up and you have a great improvement. Well, thank you very much for your question.
Thank you, Professor.
And goodbye.
Thank you for accepting our invite. It was delightful.